Hello students, today we are going to discuss in detail about the methods of gene transfer in plant. It is an method of gene transfer is an technique that is effectively introduce foreign gene into genome of the target cells. There are various methods are involved. That is the direct method and the agrobacterium mediated tumefacial methods. And apart from this, we make use of physical methods as well as chemical methods. The direct gene transfer mainly relies on the introduction of foreign DNA into the plant cell with the help of a particle bombardment, electroporation, ultrasound. All these are physical methods. In addition, we make use of the chemicals like polyethylene glycol PEG and liposomes mediated transfer of DNA to the plant cell are taking place. That is chemical method. Further, we make use of the Agrobacterium tumefaciens containing Ta plasmid. Agrobacterium tumefaciens is a bacterium that infects a plant cell and causes crown gall disease. It is mediated by Ta plasmid. Ta plasmid is found in the case of the plant cell. The Ta plasmid is called a tumor inducing plasmid. So this Ta plasmid is made as a tool or vector for the introduction of the gene into the plant cell. So the agrobacterium mediated transfer are based on utilization of the agrobacterium, a pathogen which is of dicotyledonous plants, and transfers the gene into the plant genome. So the general approach of genetic engineering plant it is outlined and follows. That is introduction of gene of interest into the cell of concerned species, and we can integrate this gene into the nuclear or organelle genome of the plant cell, and we can make the transferred gene to express the regeneration of the whole plant from the genetically modified cells are made possible. The transmission of the transferred gene to the further progeny, that is the next generation, is made possible by these methods. Let us see the expression and integration and expression of the gene, how they are stable to be transmitted for the sexual process of plant fertilization. These are all the various plant transformation methods. One is physical, chemical and biological methods. The physical methods involves the microinjection and pressure oriented biolistic or gene gun cloning method by using particle bombardment, in which the genes are coated with the particles. These particles are shooted with the help of this gene gun over the particular plant. So along with the gold and tungsten particle, these genes are transferred to the plant by this bombardment. Another one is electroporation techniques, in which we alter the permeability of the membrane of the plant cell, thereby the gene gain entrance into the plant cell. Another one is silica or carbon fiber oriented methods, in which a plant's foreign gene is inserted into the plant cell. Another one is laser mediated methods are also followed. But another one is chemical methods. Already I told you that using our polyethylene glycol and liposome mediated transfer, calcium phosphate that alters the permeability of the membrane, that is allowed to in uptake. The plant cells are allowed to uptake the foreign DNA after the alteration. Then artificial lipids and proteins and dendrimers are utilized. The biological method is nothing but with the help of Agrobacterium tumefaciens as well as AI rhizogene, and some of the viruses too involved in the transfer of foreign gene into the plant cell for this purpose. Electroporation. What is electroporation means? By exposure of the plant cell to a brief period of high voltage of electricity. That is there are two types of voltages that are induced by the electroporation. That is low voltage about 300 400 volts for about 10 to 50 milliseconds, and whereas high voltage and short pulse method, that is involving 1000 to 1500 millivolts per centimeter for 10 microseconds. This is the shorter period. Higher voltage is for shorter period and lower voltage is done for longer period. So the transformation frequency are increased by several folds. The heat shock to the protoplast prior to the electroporation, that is 5 minutes at 45 degrees centigrade, and the presence of low concentration of polyethylene glycol during the electroporation technique. So here we can see the diagram, the gene of interest that is incorporated into the DI plasmid, that is plasmid, and it is added to the culture cells. We will make the plasmid as well as the DNA, that is culture cells, are mixed together, and when the electric voltage is applied, automatically it alters the permeability. The cells try to uptake the DNA through the holes in the membrane so that the DNA enters into the nucleus. Then how to select whether the cell is having that plasmid containing DNA or not means by selective expression, stable expression of the particular plasmid containing foreign DNA, we can able to identify whether the gene is present or not. So this method is advantageous. Some plants are polyethylene glycol sensitive. Such cases we can make use of this electroporation method as the fine choice for the transformation technique. And application, it is widely used in transforming cell lines and plants of several species like tobacco, maize, rice, wheat, etc. In tobacco, the frequency is as high as 2 to 8% in the presence of 7% polyethylene glycol. The next method is microinjection. Microinjection, from the name itself, you can able to identify that the foreign DNA is inserted into the DNA of the plant as well as the animal cell. The most reliable choice for the DNA transfer in animal cell is microinjection when compared to the plant cell. Here how it is made possible means around 0.3 ml of DNA of interest is taken and is injected to the a point above the tiller node until the several drops of the solution comes out of the drop of inflorescence. What is the time of injection to be made means nearly 14 days before the meiosis, the gene is transferred to the particular cell. The method is successful and it is used for the protoplast of tobacco, alfalfa, etc. A fine tip is used, that is 0.5 to 1 micrometer glass needle or micropipette is utilized for the holding up of the particular cell. Then a fine needle is inserted containing DNA of interest for this purpose. Once it is injected, the cell is allowed to undergo meiosis. This method of gene transfer is used to introduce the DNA into larger cell, normally performed under special optical microscope for viewing of the cell and to hold the particular cell with a micropipette and to introduce the DNA solution by the microinjection procedure. This process is frequently used as a vector in genetic engineering as well as transgenetics to insert the genetic material into the single cell. That is in vitro fertilization in the case of, that is IVF methods are in the animal tissues are done by this microinjection method. The process is applicable for plant cell as well as animal cell, but it is most common in the case of the animal cell. As I told, IVF, in vitro fertilization, is done with the help of this method only. It is ideally useful for producing transgenic animals very quickly. Now the embryonic transfer or gene transfer is done with the help of this in animal cell. In the case of the plant, the DNA is injected to brassica alfalfa crops. What are all the advantages and disadvantages in the case of the micro injection? The technique does not require any protoplast, and the instruments are very simple and cheap, and it provides useful gene transfer into cells which do not generate from the cultures very easily. Technically, it is simple. And stable integration of the DNA is taking place when compared to other methods. This method is effective in transforming the primary cell as well as the cells established in culture, and the DNA injected in this process is less extensive modification is taking place, but limitation less efficient and less specific, but frequency of transformation is very low. But for this method we require a skilled person for the injection to be carried out. So skilled persons are necessary for holding the nucleus and as well as for the injection of the DNA by this micro injections method. That is, the same is applicable in the case of in vitro fertilization of the DNA into the animal cell. Embryonic cells are preferred for manipulation because it is easy to inject them DNA by this embryonic stem cells. Embryonic cells. The knowledge of mating time and oocyte recovery is very essential for this method, and this method is useful for protoplast and not for the cells with walls. Another one is my biolistics, or we call it as microprojectile for DNA transfer. Biolistic or microprojectile means the gene is coated to the bullets or ball-like structure, then it is shooted over the plant cell or the animal cell. This is by the particle bombardment by the physical method, by a microprojectile to deliver the DNA or the molecule into the intact tissues and cell. This simplest method of direct introduction of therapeutic DNA into the target cell is taking place. It was discovered by John Stanford at Cornell University during 1987 for the production of the commercial genetically modified crop, especially maize containing Bt cotton gene. What is Bt cotton means? A special bacterium called as Bacillus thuringiensis. That is a bacterium that produces a toxin called as Bt toxin, which is capable of restricting the attack of a pesticide, pest and insect. So this Bt toxin is introduced by this biolistic method. That Bt toxin was isolated from this bacterium, then it is coated to the bullets or biolistics, and further these bullets are targeted over the plant cell, root section or embryo and seeds for further procedure. The gene gun is delivering literally fires the DNA into the target cell. So the DNA is transformed by coated over the microscopic beads made up of either gold or tungsten bullets. Here gold or tungsten bullets are coated with our gene of interest. The coated beads are then attached to the end of the plastic bullets and are loaded into the firing chamber of the gene gun. As the explosive fires the bullets down the barrel of the gun towards the target cell, thus beyond the end of the barrel. When the bullet reaches the end of the barrel, it got stopped, but the DNA coated beads are continues to move forward in the target cell. Some of the beads are passed through the cells into the cytoplasm of the target cell. Here some beads are directly enter into the nucleus. Once inside the target cell, the DNA is solubilized and expressed. So helium, for the piston to work in the case of the gun, helium power or gun is used, or some of them are handheld guns are available which can make use of the helium pressure 1000 psi, that is pounds per square inch. Here we can see the diagram of the piston plunger, how it plunges and how the before and after. Before it is present, then after loading of the bullets, microprojectiles about 2.5 centimeter in diameter and 0.06 millimeter in thickness, that is the gold and as well as the tungsten at gold at 4 degrees centigrade and tungsten at the minus 20 degrees centigrade are coated with the DNA that are acting as a carrier of DNA that are dissolved in ethanol, and they are filled up. The suspensions are loaded and they are allowed to coat on the surface of the microprojectile. The microprojectile microparticles varies from 0.5 to 2 micrometer. The particles are then loaded and the piston is pressed over it. The plasmid DNA then, which is attached to these microcarriers, are then targeted to the particular cell. The DNA bound to the microprojectile are then impact tissues are immobilized with highest speed. Here we can see that the firing pin, how it pushes it and how the particles reaches the target tissue through the particular gun. Here is a diagram. It's the largest form of it. It's not handheld. The cloned genes are then added to the microprojectile, that is tungsten microparticles. This is taken and it is loaded to the particle gun, the piston, the plunger, the firing pin and a blank recharge. The plastic bullets are then loaded automatically. The vents are moving and we can see the target cells are kept at the bottom. When it fires automatically, it shoots the particle into the plant cell. Here we can see the plant cell are added with the particular microparticle that enters in the cytoplasm of the particular cell. Then when the cells are taken up and when it is planted, it regenerates into the plantlets. What are all the advantages and the limitations of bioholistic method? Normally, the advantage of this method, it requires requirement of protoplast is not necessary in the case of this bioholistic method or the gene gun method. The walled intact cells can be penetrated. Manipulation of genome and subcellular organs can be easily achieved by this method. But disadvantages or limitation, only shallow penetration of the bioholistic particles takes place and it is associated with cell damage when these gold or tungsten bioholistic particles enters into the target membranes. It may break the membrane. The inability to deliver the DNA symmetrically. The tissues incorporate the DNA to able to generate. The equipment is very expensive when compared to the handheld gene gun. General application of this bioholistic method. Normally it is successful in transfer of gene into soybean, cotton, spruce, sugarcane, papaya, sunflower, rice, maize, wheat, tobacco, etc. The genomes of the subcellular organs can be accessible by this genetic manipulation by this bioholistic method. This method is also applicable to filamentous fungi and yeast mitochondria. The particle gene gun is used with the pollen, early stage embryoid, meristem and somatic embryo. By this method, we can transfer the cry gene from Bt into maize for the resistance to European corn borer, and bar gene is inserted to the rice for the resistance to herbicide, and in the case of tobacco 1% transformation has been obtained. The next method is nothing but liposome mediated gene transfer. The liposomes mediated transfer method involves bilipid layer. It is a bilipid layer, phospholipid bilipid layer will be present. The inner core is hydrophilic and outer core is hydrophobic in nature. The sphere of lipid bilayer is transported to the cell membrane directly, and it is associated with cationic lipids that are positively charged, used for the transfer of the nucleic acid in an intact form. Because these liposomes can directly fuse with the membrane, and that will allow the entry of the DNA that is wrapped with it into the cytoplasm of the particular plant cell as well as animal cell. The cationic liposomes produced by the number of cationic lipids like DOTAP and DOTMA. These commercially available lipids are sold as a transfecting agent as lipofectin. The liposomes are now added with initially the DNA molecule, and further the liposomes are ready to add it to the target cell. The germline genesis is possible with the help of this liposome mediated transfer, and embryonic cells have been successfully transformed with our liposomes. Here we can see from the diagram the lipid solution and the DNA, and when they mixed up together it forms a lipid bilayer. Within the bilayer we can see the DNA is wrapped around it. When the DNA is encapsulated inside the liposome, then it is easy to adhere to the cell membrane, and the bilayer will attach with the membrane and fuses, and the wrapped DNA then is easily delivered into the target cell. It reaches the nucleus further. The foreign DNA is further expressed by this method. The advantages of this method is very simple and long-term stability. There is no toxicity, and production of the nucleic acid is also possible without any degradation. Another method is involving the chemical called as polyethylene glycol mediated transfection. This polyethylene glycol is a chemical that is stimulating endocytosis process whereby the DNA gets uptaken, and protoplasts are kept in this solution containing polyethylene glycol for this method. And calcium chloride is also added, and sucrose glucose act as osmotic buffering agent. After exposure to the of protoplast to the exogenous in the presence of polyethylene glycol and other chemicals, the polyethylene glycol is removed, and the intact cell and protoplast then allows the movement of these DNA as well as other along with the sucrose as well as glucose to inside the cytoplasm. Here we can see that protoplasts are suspended in a medium rich with Mg2 plus ion. The plasmid linearized plasmid DNA contain the gene of interest. Then we are adding 20% polyethylene glycol at a pH of 8. The heat shock for about 5 minutes at 45 degrees centigrade, then transfer to the ice. The DNA transferred is done with the other method called as DAE. DAE dextran is another chemical that is used for the transfer of DNA into the plant cell as well as animals. This polycationic molecule is having a higher molecular weight, and it is convenient to transfer the cells. Convenient to transfer the DNA. Further, we have DAE dextran treatment is coupled with dimethyl sulfoxide, and a shock is given. Up to 80% of the transformed cell starts expressing the gene of interest by this method. And the treatment with chloroquine increases transient expression of the DNA, and this is more successful and more cheaper when compared to the short exposure to the calcium phosphate. Calcium phosphate mediated gene transfer, that is, the isolated DNA is treated with the cells which are to be transfected in the presence of calcium chloride or potassium phosphate. Under the condition, the calcium phosphate is formed. The cells are then incubated with a precipitated DNA solution, During this method, what happens? The calcium phosphate 1 to 2% that alters the permeability of the cell so that the DNA gain entrance by this treatment method. It creates pores on the surface of the membrane, thereby the DNA gain entrance. What is the limitation means the frequency of integration of the DNA into the cell is low. The integrated gene undergoes some substantial modification. Many cells do not like this solid precipitating method adhering to them, and due to this limitation, somatic transfer of the DNA is less limited in this case. Here we can see the calcium phosphate solution. The DNA is precipitated onto the surface of the cell and a monolayer of the animal cell is seen. And the biological method which is involved in the transfer of the DNA is with the help of this soil bacterium called as Agrobacterium tumefaciens. It's a gram-negative rod-shaped bacterium. There are two types of biological bacterium which are available apart from Agrobacterium tumefaciens. Another one is Agrobacterium rhizogene. The Agrobacterium tumefaciens produces a disease in the dicotyledon plants is nothing but crown gall disease. It is economically important disease of the many plants, particularly grapes, walnut, apples, and roses. This Agrobacterium is a highly pathogenic bacterium that causes infection into the plant at the wounded site, and it produces a disease symptoms in which the wounded site by the chemotoxins many chemicals are reaching the infectious site of this bacterium, such as sugar and phenolic molecules are released from the damaged plant cell. This crown gall formation depends upon the presence of this Ta plasmid in this bacterium. This Ta plasmid, as a part of it, it is transferred to the plant cell and gets attached with the genome of the plant. This tDNA now responsible for the production of two hormones in the plant, that is auxin and cytokinin. This biosynthesis of the novel plant metabolites and opines and agropines, in addition, that enhances the plant cell to proliferate in a multiple level to form a tumor like structure called gall. So it is called as gall. This tumor gall proliferation by the opine derivatives of amino acids called agropines is responsible for it. The energy which is needed for the formation of this is in turn provided by the plants. Here is the structure of the bacterium, Agrobacterium tumefaciens. Here is a plant, Ta plasmid. The size of the plasmid is 200 kilobase pairs. Only tDNA gets incorporated into the host plant genome, only 24 base small fragments. You can see that T region, left region as well as right border of this particular tDNA, and virulence gene which is responsible for causing a virulence infection, and opine catabolism that is done by the gene opine gene, and a region which is responsible for conjugative transfer is also shown here. Here is a molecular mechanism involved in tDNA transfer. The DNA, when it infects, the tDNA alone gain entrance with the help of energy. The wounded plant fall cell in turn attracts the opine molecules which are released by the Ta plasmid are responsible for the attraction of phenolic compounds. Then virulent region of these VirE2 interacting with the protein VIP1 and VIP2 The maturity complex then moves from the infected site to the interior of the cytoplasm. Then it binds with the RAN like a protein complex. Then VIR-D2 interacts with the proteins CIPA and ROC PP2C. Then further these gain entrance into the nucleus of the molecule for the production of the phenolic compounds. That initiates the opine molecules, further aggravates the tumor formation in the plant cell. Initial infection 0. After 3 days and 18 days, we can see the crown gall tumor by the integrated DNA. This is the images of the crown gall in various plants and trees. The size is bigger and bigger. So using this method, we can able to add our gene of interest. The tDNA is normally ampicillin resistance in nature, whereas the tDNA consists of the following regions, that is two genes, TMS and AMS2, a Shuti locus gene, tumor locus and root locus, tumor locus and rooty locus, Shuti locus and rooty locus, which are capable of producing the two phytohormones, that is indolestic acid, auxin, isopentyl adenosine monophosphate, actinium. These gene coding enzymes are responsible for the synthesis of phytohormones that are gets incorporated into the plant genome for the synthesis of the production of crown gall. Further, it produces a variety of opine, which are in turn derived from arginine as well as pyruvates, which are actopine and opalin. These two opine compounds further aggravates tumor formation, which are encoded by the tDNA. Actopine and opalin the viral gene which is present in the virulent form of the tDNA is organized into 6 operons, where A, B, C, D, E and G, out of which 4 operons except A and G polycystonic in nature. Vir, A, B, D and G are absolutely required for virulence. The Vir-G locus is expressed at low levels in the vegetative cells, but high expressed in the wounded plant tissue. Once it is infected, the cells attracts, probably it infects at the region of the chemoreceptor, which senses the presence of phenolic compounds, that is exudates of the wounded plant such as acetosyringone and beta hydroxyacetosyringone. The signal transduction further proceeded via the activation by phosphorylation of Vir gene, then which in turn expresses the other virulent genes. Here what we do is we isolate the gene of interest for the production of the transgenic plant. The DNA segments are added to the TA plasmids and further added with the selectable markers. Once the genes constructed and introduced into the plant cell, we call it as transformation. The selected transformed cells are allowed to grow in a petri plate or in culture flask as plantlet, and further it is regenerated into the whole plant. Here we can see an example. A disc of the plant is taken, which is cut out from the leaf, then further it is added to the agrobacterium. Further these cells are grown as callus and later it is converted to the plant. The regeneration of the plant is then made possible and it is easy to detect. What are all the various methods by which we can able to identify means by antibiotic sensitivity we can able to identify whether the cells has uptaken our gene of interest or not and transformed. And further we can detect by them by PCR method. The DNA can be detected by after isolation of the DNA from the particular cell, and we can go for PCR modification and by southern blotting techniques or hybridization technique, where we can say that our gene of interest is introduced to the particular plant. So what are all the approved transgenic plants which are available in the market is nothing but soya bean, corn, cotton, sugar beet, squash, tomatoes, tobacco, carnations, potato, flax, papaya, chicory, rice and melon. Now, greater than 99% of the plants transgenic crops are either herbicide or insect resistant in nature, only less than have other traits. So, by using this Agrobacterium tumefaciens as a biological tool where we can genetically modify many of the products, even they we have introduced bacterial successfully the gene transfer to BT corn as well as BT cotton. It's a most approved method in the world for a successful transfer of the DNA by recombinant DNA technology. But monocotyledon plants are not very susceptible to this bacterial infection. The successful transfer is done only in the case of dicotyledon plants. That's all. Thank. The transformed plants are successful in to meet the demand for the food, energy, medicine, shelter, and clothing, and cleaner environment. The gene transfer technology is useful to produce the plants which are capable of um, hyperaccumulating toxic metals from the soils and water, thus contributing to the cleaner environment. And we can see increased in plant production so that we can meet the demand for the increasing population for the food by breeding. That's all about the gene transfer and tissue culture technology. Thank you.